Okay. And we also know that body fluid compartments are very important because they help in metabolic reaction. So for all those processes of metabolic reaction to occur, they need a medium. So this medium, it is the same body fluid. These are the same body fluid compartments that tends to help. And they also help in the texture of tissues. So we know that water and basically the same fluids, they help in the texture of tissues, the shaping of the same cells that makes up the tissues. So you find that if you put a cell in a fluid that is, let's say, hypertonic, you're going to see a reaction. You are going to see a reaction in which the water molecules will start moving from the cell, getting onto the surrounding space on outside of the cell, and the cell will shrink. If you put it in an hypertonic solution, that is a fluid that is containing low solute levels, you are going to find that water will start moving from the surrounding of the cell, getting to inside of the cell. So basically, that is just an environment that tends to affect the way the tissue is. So that is one of the roles of body fluids. They tend to help in texture of tissues, like maintenance of the body tissues and the cells and so on and so forth. They also help in the regulation of temperature. So we know that the same body fluids, specifically water, is very important in the regulation of body temperature. So let's now try to dig in, or rather let's dig in now into what the body fluid compartments are. So body fluid compartments, we say that this is just a body space in which water and dissolved substances are distributed. So one thing that you have to know is that in human beings, the total body water tends to vary. So you're going to find that there are human beings who has a lot of body water and those who have less body water. This is because you're going to find that in adults, specifically male, you're going to find that the body water, or the total body water, constitutes about 60% of the body weight. And in female, it tends to differ, which ranges from 50 to 55%. So you're going to find that in adults, you have more body water, or rather you have more fluids and more water compared to female adults. This is because in female, they tend to have a lot of adipose tissue content. So because of that, they have less body water compared to adult male. So I guess I believe we are following. So the body water is actually the amount of water that is present in the body, including the fluids that is going to be outside and inside of the body, of the body cells. So we are saying that this body water, it is going to be the water that is found outside and also inside of the cell. And as I mentioned, you're going to find the total body water consists about 60% of the whole body weight in an average adult who is weighing about 70 kg. That is an average adult. So if an adult is weighing about 70 kg, you're going to find that 60% of this weight is the body water, the total body water. So if you say 60% is of the total body water, it is calculated to be, the total body water is calculated to be 42%, or rather 42 liters, and it ranges between 42 liters to 40 liters. In some books you find 40 liters, some books you find 42 liters, but it is within the same range, between 40 to 42 liters. And females, they tend to have less body water because of the presence of adipose tissues or fatty tissues. Because you know that the fatty tissues, they tend to store less, and in fact they are not hydrophilic, but they are hydrophobic. And one thing that you also have to know is that in infants, they have less body fat and they have more muscle. So we are going to find that in infants, they are going to have more body water, which is going to be somewhere in the ranges of 70 to 75%. Yeah, so this is because they have less body fat and they have got more muscle. And the other thing that you have to know is that the rate of metabolic reaction in infants, it is quite high. And we know that these fluids, they create that medium that allows the transportation, and also that helps in the same metabolic reaction of the body. So that also plays a role. So you're going to find that infants, they have got more body, the total body water, it is actually more compared to adults. So the body fluid compartments are divided into two many compartments. So body fluid compartments are divided into two many compartments. So we have the first one, that is known as the extracellular fluid compartment. Then the second one is known as the intracellular fluid compartments. So these are the many compartments of which these body fluids are divided into. So the extracellular fluid compartment, basically this just constitutes the fluids that is found on the outside of the cell. That is why it is known as extracellular, meaning that outside of the cell. So basically, extracellular fluid compartment is just a compartment that constitutes of the fluids that is found on the outside of the cell. And you're going to find that it also constitutes about 20% of the total body weight. So the extracellular fluid compartment consists about 20% of the total body weight. So taking that, we are looking at an average weight male of about 70 kg. So meaning that if you want to know what is the amount of the extracellular fluid, we're just basically going to say what is 20% of the total body water that we are having. So we are going to say 20 divided by 60. Remember, we are dealing with 60% because we say that the total body water contributes about 60% to the total body weight. So we are going to say 20 divided by 60 times the total that we are dealing with, that is 40. And in this case, we are going to get 42 liters. So we are going to find that this whole thing, when you do the math here, you are going to find that the extracellular fluid is about 14 liters. Yeah, it is about 14 liters. So meaning that the extracellular fluid, that is the fluid found outside the cell, is about 14 liters, and it consists about 20% to the total body weight. Yeah, I believe we are following. So the extracellular fluid, the same that we are talking about, it is actually divided into two. So we are going to have this as our extracellular fluid, and this extracellular fluid is going to be divided into two parts. So the extracellular fluid is basically divided into the extravascular fluid as well as the intravascular fluid. So we are going to have the extravascular fluid, as well as this side, the intravascular fluid. So when I say the extravascular fluid, is basically the fluid that is found outside the vascular system, which is the vessels, the blood vessels. Yeah, so meaning those fluid that is being found outside the blood vessels, it is known as the interstitial fluid. Yeah, and this one tends to constitute about 75% of the extracellular fluid. It is going to be the extravascular fluid. And the same extravascular fluid, it constitutes about 15% to the total body weight. So the 75% that it constitutes, it is only from the extracellular fluids. And the 15%, it is only from the total body weight. So meaning that if you do the math, you do that, what is now the 75% of the total extracellular fluid that we are having? You're going to find that you have 10.5 liters. So meaning that of the 14 liters of extracellular fluid that we have in the body, 10.5 liters, it is extravascular fluid, which is the interstitial fluid. So basically, the interstitial fluid is basically the fluid that is found in between the same cells, the body cells, outside the vascular system, in between the body cells. Yeah. Then the intravascular fluid, this is basically the fluid that is found in the vascular system. So you're going to find that the fluid that is found now in the blood vessels, what are these fluids? So basically these fluids, we can call them, basically it is just plasma, blood plasma. Blood plasma. So blood plasma, it is actually a portion of blood, or it is a fluid which is a portion of blood, and it consists about 25% to the total extracellular fluid and about 5% to the total body weight. Yeah, so if you do 25% of the 14 liters that we have, it is going to give you 3.5 liters. And if you look at 10.5 liters plus 3.5 liters, it is going to give you the total 14 liters of the extracellular fluid that we have. Then there is also another special type of fluid that is basically in small amount. This is also known as the intracellular fluid. It is also considered to be part of extracellular fluid, but sometimes in some books the reason why you might not see it, it is negligible, is because it is very small. It just consists about 1 to 2 liters, that is, of the extracellular fluid. So it is considered to be part of the extravascular fluid, and basically this transcellular fluid, it consists of fluids that we know to be the cerebrospinal fluid that is found in the ventricular system of the brain, then also the synovial fluids, those that are found on the joints, the synovial joints, the pleural fluid, so on and so forth. That is what is constituted, or rather what is found in the transcellular fluid. So it is actually a smaller portion of the extracellular fluid compartment. Yeah. So if there is any question, you are free to comment, you are free to ask at this moment in time. So this is the extracellular fluid. So just a quick recap. Extracellular fluid compartments, this is a compartment that consists of the fluid that is found on the outside of the cell, and it consists about 20% of the total body weight, of which, if we say the 20% of these 42 liters that we took as an example, we are going to find that it will give us 14 liters. Then it was divided further into two. We are going to have the extravascular fluid and the intravascular fluid, which is under extracellular fluid. The extravascular fluid is the one that was having the interstitial fluid as well as the transcellular fluid. So the interstitial fluid, it is consists about 75% to the extracellular fluid in total, and the intravascular fluid, it was constituting about 25% to the extracellular fluid, of which, when you do the math there, we are getting from the extracellular fluid, or rather the extravascular fluid, we are getting about 10.5. Then from the intravascular fluid, you are getting 3.5. So this is the whole data that you need to know on the extracellular fluid. Then on the part of the intracellular fluid, basically the intracellular fluid is basically the fluid that is found now in the cells, or in the body cells. So this fluid, it is not found outside the body cells, but it is found on the inside of the body cells. Yeah, so it is going to be found on the inside of the body cells. Inside that body cell, that's where this fluid is counted to be. So this intracellular fluid, just from the same name intra, you can know that it is inside. So intracellular fluid, it is a fluid found inside the cell. So it consists about 40%. This one, it consists about 40% of the total body weight. And if you check 40% plus 20%, it will give you now the total 60% that the total body weight contributes to the total body weight. Yeah, so it consists about 40% to the total body weight. And if you say what is now the 40% of the example that you are having, which is 42 liters, you are going to find that approximately it will give you 28 liters, which is making sense, because if you get 28 liters plus this 14 liters, you are going to get back your 42 liters. So what you have to know also about the intracellular fluid is that this intracellular fluid, it is very important, for it helps the cells to carry out the metabolic reaction, just like the way we started at first to say these body fluid compartments containing the fluid. They are very important because they help they help in body homeostasis, the regulation of body temperature, and also the metabolic reactions of this that goes on in the cell. They also tend to help in the maintenance of the cell shape, just like the way we talked about. Like if you put your cell in an hypotonic solution, isotonic solution, you find that the cell will be reacting depending on the fluid compartment and the type of fluid that you put it into, depending on the amount of solids that is in that particular fluid. Yeah, so they also help in enabling enzymatic reaction. So also enzymes, they require a certain amount of, or rather they require a certain medium that is constituted of the same fluid in order for them to carry out their functions, or in order for them to work effectively. Yeah, so these are the main components or the main divisions of the body fluid compartment. So we have the extracellular and the intracellular, as you have heard. So right now, if there are any questions, please, you are free to ask your questions, you are free to comment. Okay, so one thing that you also have to keep in mind is that the same fluid compartments that we have, the extracellular and also the intracellular fluids, they are separated from each other by a cell membrane. This cell membrane, it is actually highly permeable to water, and it is selectively permeable, or rather it is not permeable to most of the electrolytes. So meaning that these fluid compartments that we are talking about, in as much as they allow the movement of water from this compartment to the other compartment, there are other things that does not just pass freely, just like the way we talked about transportation. So you are going to find that there are contents that the intracellular fluid compartments has, and also the extracellular fluid compartment has, apart from the water molecules. So for example, when you talk about the content, when you talk about the contents in terms of electrolytes, yeah, so we're going to find that sodium, which is Na plus, extracellular fluid here, then intracellular fluid. Okay, let me use the other part. Okay, please. Yeah, so let's talk about the content. So let's talk about electrolytes. Yeah, so here we're going to have the extracellular fluid, and here we're going to have the intracellular fluid. Yeah, so when you talk about sodium, so sodium, it is an electrolyte that is very important in the human body because it helps in a lot of functions and reactions that takes place in the body. So you are going to find that sodium, it is going to be in higher concentration in extracellular fluid compartment. It is approximately about 142 milli equivalents. So these milli equivalents, these are basically the units that are used in measuring these electrolytes. Yeah, and on the outside, you're going to find that the amount of sodium is less. It is just on the inside of the cell, which is the intracellular fluid. It is about 10 milli equivalents per liter. And then when you talk about potassium, you're going to find that it is low on the outside. It is just about 4 milli equivalents approximately, and on the inside, which is the intracellular part, it is about 140 milli equivalents. So we're going to find that these electrolytes, they tend to differ in terms of their concentration. Yeah, so you're going to find that there are those that are more on the extracellular fluid compartment, meaning that they are more on the outside of the cell compared to the inside of the cell. And we're also going to find that there are those that are going to be more on the inside of the cell, which is the intracellular fluid compartment, compared to those that are on the outside of the cell. Like when you talk about chloride, you're going to find that it is more on the outside, about 103, and on the inside it's just about 4. And also when you talk about bicarbonates, you are going to find that it is more on the outside, which is about 28, and less on the inside, which is just about 10. So there are just a lot. We are going to give you the table of these things that you just have to know them by heart, because they will help you as you progress, as we are going to progress learning more in physiology. Yeah, so we are going to find that these electrolytes, they are going to differ in terms of their quantity between these two fluid compartments, and you know the reason why it is like that, because it is very important. Yeah, so having known that, you are free to ask questions. Yeah, you are free to comment. Yeah. Any questions so far? Oh, yeah. You are free to ask. Okay, so the other thing that we also have to look at is the transport across the same fluid compartments. How do these substances move? So I'm not really going to focus on those that you did, like you did the diffusion, the passive, and also the osmosis, we know, because it involves the movement of water molecules across the semi-permeable membrane from a region of lower solute concentration to that of higher solute concentration. So the other thing, that the most important thing that I want to highlight on the transportation across this, it is what we call the Starling forces. So they are what we call the Starling forces. These forces, they create a pressure that tends to facilitate the movements of this fluid across the capillaries or across the blood capillary wall. So these Starling forces, they are actually very, very important in the movement of these fluids. So they facilitate the movement of the fluids between the intravesicular as well as the extravesicular, just like we talked about. So the Starling forces, we have what we call the hydrostatic pressure. Hydrostatic pressure, noted as the HP. And we also have what we call the oncotic pressure. The oncotic pressure, that we can note down as the OP. So basically how this works is that the hydrostatic pressure, it is the pressure that is involved in driving out the fluids from the capillary into the interstitium. So meaning that if this is a blood vessel that we are having here, let's say this is a blood vessel that we are having here, the same capillary that we are talking about, this side we are going to note it as the arterial blood that will be moving from the artery, and this is going to be the venous blood that will be flowing in the veins. So what happens here is that when blood is flowing from the arterial going into the venous, what happens here is there's going to be an exchange of this blood, because we know that the arterial blood is rich in oxygen. So for it to transition to go to the venous one, it means that the oxygen that was in that arterial blood has been used by the body tissues, and the blood has to flow back to the heart so that it can be oxygenated. We will know the flow when the blood goes through the veins and gets into the heart, and the heart to pump into the lungs, and purification there, then addition of oxygen, then comes back, and so on and so forth. So one thing that I just want to highlight here is that when blood is moving from the arterial, you are going to find that it is going to have these two pressures present. So there is going to be the hydrostatic pressure as well as the oncotic pressure. So basically the oncotic pressure is the pressure that is going to be formed because of the presence of plasma proteins. We know that in blood plasma there are proteins that are found there. So these proteins, they create a force, or rather they create the same force, which is known as the same force rather, which is the oncotic pressure. Yeah, they create this pressure called the oncotic pressure. So this oncotic pressure, it is the one that is going to facilitate the retaining of the fluid. It does not support the fluid from being pushed outside. No, what it does is that it tries to keep the fluid just inside here. So when blood is coming from the arterial section, you are going to find that it will come with a lot of pressure. So meaning this hydrostatic pressure will be high, and the oncotic pressure is not going to be as high as the hydrostatic pressure. So it is going to be a bit lower or even low. Why? It is because the heart, when the heart is pumping blood into the systemic system, you are going to find that it will pump it with high pressure, so that even in the body extremities where there are very small or tiny blood vessels, there can be perfusion of blood which is oxygenated to take oxygen there and other required nutrients. So that's why it pumps with a lot of pressure. So you're going to find that blood will be coming with a lot of pressure, of which the hydrostatic pressure is going to be high, and the oncotic pressure is not going to be as high as the hydrostatic pressure. There's going to be a difference in pressure. That difference in pressure between the same hydrostatic pressure and oncotic pressure is what will facilitate the movement of these fluid from the inside of the vessel, or the vessel, going onto the interstitium. Yeah. Then as blood passes and goes on to the venous part, you are going to find that the hydrostatic pressure is going to be low compared to the oncotic pressure. Then the fluid is going to be retained, meaning that this side you are not going to see any movement of the fluid from the intravascular section into the extravascular section. Why? Because the hydrostatic pressure that is facilitating that movement is low here. But on the arterial section, we are going to find that the hydrostatic pressure there was higher compared to the oncotic pressure. So because it was higher, it was facilitating the movement of the intravascular fluids onto the interstitium, which is the extravascular fluid. So these Starling forces, basically, they are very important, and these are the roles that they play. And when we start also looking at renal physiology, we'll also show you even more in detail on how these they operate. But for now, let's just try to keep that in mind. And basically, this is what we need to know, and it will help us to answer the follow-up questions that we are going to show you. Any questions, any comments so far? Okay, so upon knowing these few things that we have looked at, we also have to know that the same body fluid compartments, we can measure them to try to know how much body fluids, we are having. So basically, scientists, they had to sit down to say, how are we going to measure these body fluid compartments? How are we going to measure these compartments to know how much fluid or how much volume of fluid we are having in a particular compartment? So they came up with methods, of which one of the methods is known as the indicator dilution method. So this indicator dilution method, it is a method that involves using an indicator. So indicator, these are just basically substances that are used for each particular fluid compartment. So each fluid compartment, like the way we are talking about the extravascular, the intravascular, the extracellular fluid, has got specific indicators or dyes that are used to measure. So if you want to measure specific, for example, the blood plasma, which is which is the fluid, or the intravascular fluid, we have indicators like Evans blue. That's the one that is used. Like Evans blue, it is part of the indicators that are used to measure the blood plasma. So they came up with this method that is known as the indicator dilution method. So this indicator dilution method basically involves indicating, or rather, sorry, not indicating, but involves injecting that particular dye that you have into the targeted or the fluid compartment of interest. So for example, let's say you are having this indicator A. So this indicator A, you want to inject it into a particular fluid compartment of interest. So what you're going to know is that, first of all, before you inject it, you have to know the amount of this indicator that you're about to inject. Then after knowing the amount, you also have to know what is the concentration of the indicator that you want to inject. Upon knowing that, then you are going to inject this indicator into a body fluid compartment of interest, or into a compartment of interest. When you inject that, after some minutes, it can be 15, 20, 30, depending on the indicator that you are dealing with as well as the compartment, you are going to get a sample from that particular compartment. When you get a sample from that particular compartment, you are now going to measure what is the concentration of the indicator that we injected after it has diluted into that compartment. Then you are going to get the concentration of that particular indicator in that diluted fluid compartment that you have drawn as a sample. So when you get to know that, then you are going to use the formula, that is the volume of that compartment of interest is going to be the amount of that substance that was injected times its concentration divided by the concentration of A after dilution. So meaning this after dilution, it is after you have injected it into that fluid, into the compartment of interest. Then after you do that, then you are going to do the math here. Then at last you are going to calculate. Then you know the volume of that particular compartment of interest that you are dealing with. And there are also some exceptions that you need to consider. Let's say you inject this patient, and this patient that you inject feels like urinating, then they go and urinate. How is that going to affect your results? Because you have already injected your amount of your dye that you are working with. So there was an introduction to this formula that was done so that it can try to cover up that part. So what they did was, when let's say you inject a patient with particular dye in that compartment of interest, and the patient, let's say, goes and urinates, you're going to get the urine that the patient urinates, and you're going to find out what is the amount of that dye that we injected, which is in the urine. When you get that amount, then you are going to say the amount of A that we injected minus the same amount of A that was excreted. Then when you do that, you calculate, you find, and you multiply it with the concentration of A that you measured before you injected, divided by the concentration of A after dilution. Yeah. So this formula basically it can apply even as you are calculating the total body water. As you are calculating the total body water, yes, that's what we are talking about. You can use this method as we are going to show you in the question that we are having after the lecture. Yeah. So upon knowing that, they had to find that, there are certain things or rather there are certain compartments or certain fluids that also needs to be measured, and this formula wasn't quite just adding up for every part or for every type of fluid that you want to measure up. Let's say, for example, you want to measure the red cell volume. Also, you want to measure the plasma volume. Also, you want to measure the total body volume. You cannot just stick to this one formula. So there are other specific formulas to simplify things that was brought up if you want to measure them. So let's say you want to measure the plasma volume. You are free to comment, to ask the question as the video is going on. This is for us, for you, for us. So let's say you want to measure the plasma volume. So you're going to say plasma volume. That is now your compartment of interest. How do you measure it? So you're going to say, when you want to measure the plasma volume, you say so if you want to measure the plasma volume, we talked about the indicators. They say you can use indicators like Evans blue. Evans blue, like this one in terms of spelling. Evans blue, and blue is just like the same for color blue, and sometimes it is represented as T1824. So most in MCQs you are going to find something like T1824. Just know that that's Evans blue. Apart from Evans blue, you can also use what we call radioactive iodine. Yeah, you can use it to measure plasma volume specifically. Only these are just the dyes for plasma volume, if you just want the plasma volume only. So you can say now the formula, plasma volume is going to be equivalent to the amount of that dye that you are dealing with that was injected. So you're going to say the amount injected, or the amount of dye injected, divided by the concentration of that particular dye. So when you do the math there, you find your answer. That answer means that you have gotten the plasma volume of interest. I believe we are following. Then apart from just calculating the plasma volume, these fluid compartments, we can also calculate what we call the red cell volume. So basically the red cell volume, this is just the volume that is consisting of the red blood cells only. So you're just dealing with the amount of the red blood cells that you are calculating. So the red cell volume is measured, is calculated by the formula. Red cell volume is equal to the total blood volume minus the plasma volume. The reason why we are doing this, if you can relate, is because the red cell volume, since we know that just the volume consisting of red blood cells, you know that plasma is a fluid, or it is a portion of, or rather it is a fluid where these red blood cells are going to be found. Yeah. Apart from the red blood cells, we also have other cells, but the cells of interest here, it is just the red cell volume. So you are going to find that for you to calculate the red cell volume, you can say total blood volume minus plasma, because the total blood volume, when you remove plasma, the greater concentration of what is going to remain will be the red cell volume. So when you know the total blood volume, you also know the plasma volume you have already calculated, you can now then get the red cell volume. You can get what we call the red cell volume. Apart from that, we can also ask to say, now how can you get the same total blood volume, so that when you put it here, it makes sense, so that you can get the red cell volume. Yeah. So the total blood volume, basically you can calculate it by the formula, which is total blood volume is equivalent to the plasma volume. So meaning the plasma volume, the amount of plasma that you got, divided by 1 minus hematocrit. So hematocrit is also known as the packed cell volume, or the PCV. Yeah, so in some textbooks you see PCV, some other textbooks you see hematocrit, just basically the same thing. And these values, they have got the normal ranges, and mostly they give, but you have to know the normal ranges. The parameters, guys, they are very, very important, because most of the questions you'll be encountering, they'll be giving you a clinical scenario questions. Like, it will be giving you clinical scenario questions, give you the records, or rather the information about the patients. It's for you to know what is abnormal, what is normal, what is within the normal range. So these parameters that we are going to be giving you have to make sure that you know. So even for the hematocrit, there is a range that is there, which will help you. So they will usually be giving you, as I'm going to show you in the question we are going to do. So when you calculate the total blood volume, then you know, okay, so this is the blood volume that is remaining in this patient. As the patient encountered this accident, they didn't lose enough blood, or they lost enough blood, so meaning that the fluid therapy that you are going to give them is maybe 2.5 of this fluid. Then you'll be able to know, as a doctor, you just tell the nurse to say, okay, give this fluid to this person, and the volume is specified, because you would have already calculated what is the total blood volume that is remaining in this patient. Yeah, so upon knowing this information, I would like to show you some of the examinable questions that comes under this topic, and you are really going to appreciate them, because the information that you are having right now, it is accurate, and it is the answer to this question that we are having here. So basically, this is the question. Yeah. So this is the question that we are having. The question is saying that Mr. Niza was involved in a road accident and lost a lot of blood due to hemorrhage. He was quickly rushed to your clinic for medical attention. Upon arrival, the doctor requested that you carry out some tests to estimate the volume of the fluids lost before giving any fluid therapy. So 2 mL or 2 milliliters of Evans Blue with the concentration of 2000 milligrams per liter was injected into the patient, which is Mr. Evans. Then after 15 minutes, 5 mL of blood sample was collected, given to the lab technician for analysis. So the results indicated that the concentration of T1824 in the blood sample was 1.6 milligrams per liter. Now answer the following question. So if you have seen here, just from the information that you have been given in the question above, you can see that they are using an indicator which is Evans Blue, also noted down there as T1824. So you will know this indicator is basically used for this fluid compartment, which we talked about as plasma. Yeah, when you are testing for a fluid compartment containing of plasma. So you will know that here we are dealing with the plasma volume concentration, or rather the plasma volume itself, because of the indicator I told you. And the other thing that you also have seen is that they first had the volume of the indicator that was injecting, and they also knew the concentration that was indicated. So upon knowing the volume and the concentration, basically these figures are helping you to get something that I told you there. And the other thing that you also have to know, they say the question they are saying that calculate the plasma volume of this patient and estimate plasma loss if the normal should be 3 liters. Yeah, so from the information that you have there, you would be able to calculate the plasma volume. Then the plasma volume that you are going to get, you will now subtract it from the one that they have given you to say if the normal is 3 liters. So meaning you are going to subtract it there, what will remain, then that will be your answer. Then given that the hematocrit, which is the PCV, as I told you, is 45%, calculate the total blood volume. So basically the hematocrit, one thing that you have to know about the hematocrit is that hematocrit has to be in it does not have to be in percentage form. You have to take it back to decimal form. So 45%, if you take it back to decimal form, you just divide it by 100. That will give you 0.45. Yeah, so if the hematocrit was 25%, just divide it by 100, because we know that they have given it to you in percentage, and every percent is out of 100. Is out of 100. So just divide it. So given that you have the hematocrit, which is 45% in this case, calculate the total blood volume. Yeah, you can calculate, because already you're having the hematocrit, you would have calculated the plasma volume there. It's just a matter of plugging in the values and the formula we have. Then from there, calculate the red cell volume, of which the red cell volume we talked about it, and you can also calculate with the formula that was given. Then the other one was using a flow diagram, illustrate blood fluid compartments in a 70 kg man, 3 marks. So basically here what you have to do is the same basic concepts that we started with here. The same basic concept that we started with, saying that the extracellular fluid compartment consists about 20% of the total body weight, of which that was given as 42 liters. Just the same information that you have here, we are now going to put it in form of a flowchart that has been requested. So the flowchart, just to give you a sample, but the questions, the answers to the questions will send to you immediately after the lecture. So just to give you a sample, we are going to find that you have to write it in this manner. So here you are going to write. At that point, you are going to label it as the total body water that you are having. You name it here. then even put the amount. Then from there, you're going to put the division. So the total body water, or rather the total body fluid compartments, is divided into two. We are going to have the extracellular and the intracellular. So when you have the extracellular fluid, the intracellular fluid, you are going to go to the extracellular, you divide it, you have intravascular and extravascular. So intravascular and extravascular. On the extravascular, you know that the extravascular is having two special types of fluid compartments. So we are going to have the transcellular, and we also have the interstitium. Then the intravascular is basically the one that we are referring to as plasma. Then you also put figures. Oh, this one it is 42 liters. Then here extracellular fluid, it was constituting about 20% of that. So we know that it is 14 liters. Here it was 28 liters. Then from there we know that the intravascular was constituting about 10.5 liters. Yeah, so basically it was considered about three. This one was the extravascular. Yeah, that was the one considered about 10.5 liters, and then this one, the one for intravascular, which is the plasma, was considered about 3.5 liters, and so on and so forth. Then you just go down, break them down like that. So this is just the flowchart that was required for you to write there, and they'll give you the all 7 marks, and you get the all 15 marks if it is a 20 marks of the question, because basically these are the questions that you'll be encountering on the short on section B for the short essay question part. So don't worry about the answers. We are going to share the answers in the group, and feel free to comment and also tell us how you are enjoying the lectures. Feel free, actually, just feel free, communicate so that we know what we should do so that we can meet your needs and your expectation. Otherwise, thank you very much for your time. We really appreciate, and see you next time. If there are any questions, you are free to post the questions in the group, and we'll be happy to attend to you. Have a blessed evening, saints.